So you can, so the first talk is the egocentric behavior explains a significant variance in hippocampal neural data. And this is by Mohammad Yaghoubi from McGill University. Okay, Mohammed.
Bismillah ar-Rahman ar-Rahim. I'm going to start with acknowledging all of those people who have put a lot of effort to make this amazing symposium happen. I really enjoyed all of the presentation before mine, and I apologize for the inconvenience. Um, yeah, the title of the talk is Egocentric Behavior Explains a Significant Variance in Hippocampal Neuronal Data. And I'm going to start off with the idea of cognitive map. That's basically a theory which was coined by Edward Tolman in 1948. And the theory says, for example, imagine there is a mouse and the mouse is going to navigate this space to get the reward. It does some navigation and gets the reward. But if the mouse understands how the space works, if the mouse understands how the elements of the space are connected to each other, for the next time, the mouse could make a more efficient navigation towards the reward, right? So that basically tells the mouse, if you provide a map of the space, if you provide a map of the space which gives you a connection between all of the elements of the space at once, then you could provide more efficient navigation through the trajectories that you have even never gone through them before. And that was basically the idea of having a cognitive map. That was the idea, but there was no neuronal explanation for it. And it took a couple of decades when people were able to record from these deep regions in the brain, which is called hippocampus and internal cortex. Today, I'm going to just focus on the hippocampus. And if you make a cross section of this hippocampus like this, you're going to see this beautiful structure here. The top part of the hippocampus is something which is called CA1, and the bottom one of it is CA3, and this other complementary part of the structure in the top right is called dentate gyrus. So what people did is that they put an electrode in the hippocampus and they looked at the animal as the animal is moving around in a box. And what they observed was that when the animal goes to a particular location in the box, the neuron in this hippocampal region starts firing. If the mouse goes to the other region of the box, it does not fire much. And in fact, the neuron is very sensitive to the location of the mouse. So basically, this neuron is tuned to a particular location in the box. And so this blue uh, square that you see is something that we call rate map, and they show some sort of tuning curve of the neuron. And if there is a large population of neurons in which each of these neurons are coding for a location of the box, you're going to have an ensemble of neurons in which each of them are coding for a location of the box. And there's going to be a coverage, right? A coverage of all of the location in the box, and you're going to have a complete sense of the space, complete understanding of all of the location in the box. And that's basically what John O'Keefe proposed as a neuronal basis for the cognitive map that Edward Tolman coined a few decades before. But this is not the only cells which is spatially modulated. There is a zoo of different type of neurons which are coding the space in different manners. For example, in this box at the middle, which is surrounded by this orange rectangular, we see in the left side of it, we see the place cell, the cell that we just talked about it. In the right side of it, we see the landmark cells. The landmark cells are basically the cells which are sensitive to some objects which are located in some particular location in the box. In the top box, the blue box, we see some other neurons. These neurons are not in the hippocampus, they are in the internal cortex, and the internal cortex is a region nearby to the hippocampus. We see neurons which are modulated spatially, but the pattern of the modulation is more complicated. For example, these grid cells are periodically patterned, and there are these object vector cells, and there are border cells in which they respond to the location, to the space in different manners. And these discoveries brought the Nobel Prize up to 2014 for these people. Um, so now the question is that, does hippocampus only codes for space? And the answer is not exactly. So the fact that we have been seeing like spatial coding in these neurons is the fact that the tasks have been very spatially structured. In these cases, people try to design tasks in which they require different type of stimulus like tone, time, odor, and they observe that neurons in this hippocampal region, they also could code for, like, in this case, the frequency of the sound, in this case for time, and in this case for odor. So it's not just location. There could be other features of the space which could be coded by hippocampal neuron at the top of location. And to investigate this idea further, we try to design a more cognitively demanding task and see how hippocampal neurons could respond to different variables in this task. So the task is called um, delayed non-match task, sample to non-match task. And the way that it works is that we put the mouse in, we put the mouse in a chamber. There is a screen in front of the mouse which is going to be turned on. The mouse goes to that screen, pokes it. The square is going to turn off. There is a delay of 2, 4, or 6 seconds. After the delay, there's going to be two squares which both turn on, and the mouse has to pick the mismatch one in order to get the reward. So basically, when we train the mouse, we start from the day one. The mouse learns how to do the task, so the performance gets better, better, and better. But when we change the delay from 2 seconds to 4 seconds, the performance drops down, right? Because the task has somehow has changed, and we some sort of uh, reinforce some error into the task. The performance drops down, but the mouse again starts learning the task, and the performance keeps going on again, and we again change the delay from 4 seconds to 6 seconds, and the performance drops down, and the mouse tries to increase the performance again to get more and more reward. So this is about the structure of the task, and we use the miniscope, which is this little tiny thing at the top of the head of the mouse for recording neuronal activity, and we use a deep learning based software which is used for tracking basically different body parts of the animal for future behavior analysis. So one thing that we see is that the mouse performs some stereotype behaviors. Two of these stereotypes behavior are being depicted here. The blue curve and the red curve shows this pattern of navigation that the mouse goes over them very frequently. And we see that there are distinct population of neurons which are coding different phases of this stereotype task. For example, there is a population of neurons which are responding to this pattern of behavior, but it's silent for the other pattern of behavior. And there are some population of neurons which are responding to this pattern of behavior, but they are silent for the other pattern of behavior. And if you look at the uh, lower dimension neuronal trajectory uh, of this basically the time interval that this the behaviors are happening, we see that these neuronal trajectories are perpendicular to each other, which is not surprising, right? They are this completely distinct population of neurons. And the interesting thing is that we can look at the single cell level and see there are cells which are coding for different phases of this task, and we can show them actually here. So you remember what was the rate map, right? This is rate map. The mouse is, so this neuron is just responding when the mouse is up here, and the heading direction should be downward. So the mouse is up here, heading down. And as the mouse keeps moving down, we see there are different neurons which are responding to that particular location. And when the mouse turns back to the reward zone, which is up here, we're going to see there are other neurons which are responding to different locations of the mouse as the mouse is traveling upward. And basically, this population of neurons are coding for the whole pattern of behavior. So that was one observation, which indicates the neuron is not just coding for the space, but it also cares about the directionality of the neuron of the mouse, right? So, for example, this neuron codes for this location, but it's also necessary for the mouse to be heading down, right? This is the heading direction tuning curve. And let's say this neuron, pretty much the same location, but it's necessary for the mouse to be heading up. And so it's not just location, there is heading direction as well at the top of location. There is another observation that we have seen here is that when the mouse is receiving reward, like at the very top here, we see that neurons care about the fact that where the mouse has come from towards that reward. For example, if the mouse is coming from this side of the chamber and getting reward, or the mouse is coming from the other side of the chamber and getting reward, the neurons are going to be selective to the fact that where the mouse is coming from, right? So they are some sort of reward slash location cells, but they are also caring about the trajectory, which trajectory the mouse has been going through prior to the receiving the reward. So these two observations are basically suggesting that the neurons are not just coding for the location, but there are other dimensions of behavior which are important for the neuronal response. To go to the behavior analysis deeper, we try to use some deep learning approaches to develop some deep learning approaches, and the way that we do it is as following. So we go into egocentric framework of the animal, and by egocentric framework I mean uh, so we track multiple points on the mouse body, and we use two of these points to move into the egocentric coordinate of the mouse. By egocentric coordinate, I mean the coordinate which is attached to the back of the mouse. And we see the evolution of these points relative to this egocentric coordinate. In these circumstances, we don't care about the location of the mouse, but rather we care about the pose of the mouse in this new coordinate system. I hope it's clear enough. There was a movie here, which unfortunately I'm not able to play that. But if we transfer this information from allocentric coordinate to egocentric coordinate, basically we are washing out the effect of location and we are just caring about the pose of the mouse. And this is what we are going to get after this transferring to the egocentric coordinate. So we have these traces. We use a deep network, which is called autoencoder, variational autoencoder. This is vastly used in deep learning community for finding a structure in the time series. And what we get from these complicated trajectories is a lower dimensional representation of the behavior. I know it's a little bit of abstract here, but think about it like as the mouse is going through different regions of the place, right? Mouse is basically traveling in an allocentric or actual physical space, right? But as the mouse is changing his pose, we can think of it as navigating in a behavioral space, right? For example, the turning left is different from turning right, right? Or rearing or maybe compressing mouse as the mouse is compressing himself or stretching himself. These are going to be all different location or different locations in the behavioral space. And this is the way that we quantify behavior. So we can cluster this latent space of behavior and we can look at the transition between motives of behavior, right? And one interesting thing that we see is that just by looking at just by looking at the matrix of transition of motives of behavior for each mouse, we're going to be able to decode the mouse ID. So imagine you have 5 mouse, and for each mouse I have 10 sessions of recording. This pipeline of analysis enables me to perfectly decode the mouse ID just by looking at the behavior. So that means even though the behavior, the structure of the behavior is pretty much similar across species, like the same species across the animal, right? But there is enough differences from case to case in which it enables us to decode that animal ID perfectly. We can also define the rate map in the latent space of behavior similar to what we saw for allocentric behavior. And that basically is going to say, for example, if the neuron is responding to, let's say, a particular pose of the animal rather than uh, like what we saw before, where the neuron was uh, responding to a particular location of the mouse. So now the question is, all right, so we have a pipeline of analysis which enables us to quantify behavior in a very detailed and elegant way. What does in like how could we say that this information is something that neuron is caring about it? So to answer that question, we use a couple of techniques. First of all, we can use GLM. GLM is something opposite of decoding technique. In decoding technique, we look at the neuronal activity and we try to decode the uh, behavioral variable. In the GLM, we look at the behavioral variable and try to encode the neuronal activity. So what we do here is that we make a full model. The full model contains the following information: the position of the mouse, the velocity of the mouse, head direction of the mouse, and angular velocity of the mouse. And the VAME is the information that I get from this egocentric framework pipeline of analysis. And we see that like when we train the network to encode the neuronal activity, um, we can explain some variants of neuronal activity, right? And we can lesion the egocentric part and see how much of the variance is decreased. If there is a significant amount of variance of the neuronal activity decreased, that means that egocentric information has been important um, for explaining the variance of neuronal activity. So we do that and we observe that like lesioning or removing the VAME information damages the model the most, which means there is a significant amount of information which is being coded in the neuronal activity which comes from the pose and this like detailed behavior of the mouse. We can also develop some information theory approaches, which is basically, for example, if we look at the just location of the mouse. I'm
So,
sorry.
Mr. Yaghoubi, you are over your time. If you can
Oh, okay. So
wrap
maybe
up.
just a minute or two, if you don't mind.
Well, you're already 3 minutes over, but you can take 1 more minute maybe.
Sorry, yeah. So yeah, we use information theory approach to quantify that, and we see that like um, including the egocentric behavior, the detailed egocentric behavior increases the information that we see in the neuronal activity. We do the same thing in the population level, and we observe the same thing. The egocentric behavior is important. So with that, I'm going to just go to the conclusion, which is basically a pipeline of analysis. We have a pipeline of analysis, which is elegant enough to basically reveal individual differences across animals. And we also observe that these neurons code for the location, but at the top of location, there are these other aspects of behavior which are being coded as well. And I'm going to thank all of the people who have contributed to this project, especially my mentor, Dr. Mark Brandon, and all other people. And thank you all for your attention. And I'm happy to take questions if there's time.
Thank you. If there are any questions, we have time maybe for one question. You can raise your hand. And so while people are thinking, I'm just want to summarize my understanding from your presentation. So you basically used something like Deep Lab Cut to basically, and then some autoencoder, variational autoencoder to clusterize the different poses of the rats, and you found information about these pose information in the firing rates of the CA1 neurons in the rat.
Yeah,
Is that
that's
summary correct?
yeah, that's exactly it. I'm glad that you got it.
Yeah, and is this something that people have not reported before?
So that's a good question. So that's why I initially started by the idea of heading direction and trajectory dependent studies. Um, so basically, in neurons being directionally tuned has been reported in the linear track. And when people look at the two-dimensional navigation, they don't see it. But there was one study in which they trained the mouse to do some stereotype behavior in 2 dimension. And in
Uh-huh.
that case, people started saying, okay, now neurons become directionally tuned, and I think that's what is happening here as well. So the mouse is using some stereotype behavior as a strategy to solve the task.
Uh-huh.
That's one thing. The other thing is in a study from Matt Wilson lab in which like there is this W-shaped maze that you may have seen, right? The mouse like alternation task. The mouse
Mhm.
goes to the middle, but it doesn't count. It doesn't go back to the arm that he's been before. Now
Right.
he goes to the new arm, right?
Yeah.
In this particular in this experiment, they observed this trajectory dependent cells in which they interpreted that as a mechanism for the mouse to disambiguate the memory, right? Because if you are in the same location,
Yes. You
but
need to know what was your previous round.
exactly,
Right?
like depending on the trajectory that you have been before in
Mhm.
that trajectory, so they interpreted that as a disambiguation mechanism and something which is like basically trajectory dependent, which is also something that we see here. So I basically showed those things as a proof of concept. But I thought at the top of this traditionally defined behavioral variable, there might be some more elegant uh,
Okay.
aspects of
And
behavior which will be explained by neural activity, and that's what we see.
how much of the firing rate was explained by place, but by location versus by the pose? Like what part of the R squared uh, was, I mean, was it a big part of the neurons, or like let's say 80% of the firing could be explained by place and only 5% by pose, or what was the relationship between the two?
I don't have an exact number for that, but I'm sure place
I
is
mean,
going to be like the dominant factor.
so
But
you can quantify that using, you can have a different partial R squared for each of these things.
for
Uh,
sure, yeah.
right.
But something that we see here is that, like in the first session, when the mouse has not developed that stereotype behavior yet,
Uh-huh.
we see like that egocentric behavior is not that different. But as the mouse develops some
Uh-huh.
stereotype behavior, then those aspects of behavior becomes more significant. And we see like this in this purple curve, which comes from this VAME, is more like being being distinct from other behavior.
I see. I see. Thank you very much. Any other questions? Anything in the text? Uh,
I will also leave my contact information if you have any question or point of discussion. I'm more than happy to discuss or chat.
okay. Very good talk. Thank you, and